also gonna post the video that Dr. Myers posted where we worked all the way up through citrate. So I'll post that video under the modules so you can look at that video where we cover all of these, but hopefully you guys will cover all these together. Again, watch the video again once you get home. I really want you guys to do well on this. Hoping, really hoping. Okay, I want to get the highest one in all the classes. We all compare notes at the end. We're gonna get the highest one. Okay, okay, so transformation. So transformation, there were a lot of pieces to this lab, but what was the main purpose? So let's go through what was the main purpose of transformation. Everybody think about it really quick before you answer. What were we transforming? What were we transforming to and from? What did it mean? What's the point? Try and think through it a little bit for a second. So we're transforming. And this is a really cool lab because actually this was the lab where you got to actually do genetic engineering. This is what they do in a bacterial lab. This is how they produce all kinds of pharmaceuticals now. They have, I was telling Dr. Dominicus before you guys got in here, was that in our lab at Cornell, they have this room you walk into. It's like a big giant room you walk into, and just the bacteria smell is like, hits you over the face. But they have these big doors and these big wheels, and you roll, you know, have you guys seen those before, like in a library where they, like all the shelves are rollable, and you turn these big wheel and it rolls out your shelf, and then you have your course number on there, so you follow your course number, and there's all your plates, your agar plates that you've been working on. But they'll have like a big giant incubator, so it's like plates and plates and plates of everybody's work. And they try and keep them all in the same incubator. You have to check in and check out before you get in there. And, but what they would do, like in an insulin lab, where they're producing insulin, is the same kind of thing. It'd be a big room. There's agar plates if they don't use something bigger now. I think they used to have plates. I don't know if they use like larger agar situation at this point. I don't know what they use anymore. But E. coli would be producing insulin. It's like a giant insulin factory. And how they do that is they transform the bacteria to produce a gene, to produce a protein that they don't normally produce. So now they're producing insulin. So we're gonna transform. So we transform with the purpose is to transform E. coli by, what do we want to say? To accept.
Induce competency.
Yes, let's say that first. Let's say that second. So to accept
Plasmid.
a plasmid
By inducing complement.
by, yeah, you know what I wanted to do actually is try and give you guys like 1 or 2 words. I'm being too wordy. Let's just do some arrows, because you guys will, all we're doing is guessing A through E, right? So if you can guess A through E, if that helps you to identify it. So transformation, we're doing our plasmid, competency, should we say competency? E. coli.
Is the lab practical all multiple choice?
Yes, except the bonus. Heat shock. CaCl2. Bring plasmid close. Yes, to bind to lipopolysaccharide. But yeah, you could just say to bring plasmid close to the E. coli. Then we're gonna heat shock it to induce competency. So heat shock and competency was an important step. We use CaCl2 into our E. coli. So what else did we wanna do? So at the end, our results, so this is the purpose. Here's our organism. You should organize it better. I'm sorry, I'm not organizing it very well. So our methods, here's our methods. I'm so sorry. Let's do it. I don't like to be disorganized. Okay, so one, purpose, to transform E. coli. 2, organism, E. coli. 3, methods. So we're going to CaCl2, bring plasmid close. And then we did heat shock to induce, is it induce or induce? Let's say competency. Induce? Yeah. Okay, competency. Then what we did is we plated on an ampicillin plate, which we'll talk about why. And then we looked at it under the UV light. Is that everything we did, basically? We got it really, really cold, then really, really hot, then really, really cold. That was our heat shock.
Yeah, my question for this is, do you want us to be really familiar with the steps with this one? Or just understand the CaCl and then the heat shock?
Yeah, heat shock is just cold,
Okay.
hot, cold.
Yes.
That's all you need to know. CaCl2 is what binds the plasmid.
Okay.
Yeah.
Cool beans.
And then we plated it on an ampicillin plate, we'll talk about that, and then we looked at it under UV light. Okay, so for reagents, we didn't really have any reagents other than the CaCl2. 5 are results. So results were, we had GFP glowing transformants
What's GFP?
and satellite cells. Okay, so let's talk about what GFP is. So GFP is green fluorescent protein. So we had GFP glowing transformants and we had satellite cells at the end that we looked under the UV light. So let's talk about what a plasmid is really quickly. So let's bring that up over here because this is important. So I like asking this question on finals and things like that because I like that this plasmid, which is just, remember, a tiny circular piece of the bacterial chromosome. It's this tiny circular piece, but it represents the chromosome as a whole. Because we have to know what pieces this plasmid has to have. So remember, if we have an E. coli, there's this big spaghetti mess that's in the nucleoid region. And then over here, it has these little plasmids. So this is a zoomed in plasmid. It got in there, usually it'll get in there in nature because of conjugation. Because that plasmid will form a sex pilus. The sex pilus, now the plasmid will be transmitted bacteria to bacteria. But we're going to transform. That's also can be done in nature. When, say for example, this bacteria dies, all its plasmids are just circling around where there's E. coli everywhere. So those E. coli that are standing around, if they're competent, they can just take up from the environment, take up plasmids from the environment. That happens in nature also. But we're going to induce competency. So to induce competency to our non-plasmid owning E. coli, we heat shocked. So we made pores, we melted that outer membrane, and the plasmid, then we put plasmids in the soup, in the Eppendorf tubes, and then those plasmids were able to get into the bacteria. Some of them worked, some of them didn't work. Some of them died, some of them didn't die. Okay, so let's talk about what was on the plasmid. So we have to replicate the genome, what area do we have to have where DNA whole what to replicate? 3? 3. And this area is called ORI. That's the origin of replication.
Do you mean 3?
DNA pool 3.
Oh, it looks like a 4, sorry.
Oh, sorry. Sorry. There we go. Okay, DNA pool 3. There we go. You're like, you said 4. Okay. So also in a plasmid itself, you want these things called restriction sites. And these are areas where restriction endonucleases will come, these enzymes will cut. And the reason we want to cut the genome is because we want to include some sort of gene here. So an insulin gene. This is normally what we would do with the plasmid, is put our gene of choice in here, whatever we want our bacteria to do. If we want our bacteria to mimic diabetes, if we want our bacteria to mimic some other issue.
Size difference.
The size difference?
One was a small plate, one was a big plate.
Well, that's because one had antibiotics and one didn't.
Yeah, and one was, are you sure we didn't just have 2 like that?
So the controls were ampicillin negative, plasmid negative, to make sure they had growth. And then the other control was ampicillin positive and plasmid negative, to make sure that we had no growth.
Yes, so I think here we only had 2 like that.
So 2 results.
One had E. coli, here was the positive, here was the negative, right?
Yeah.
One had E. coli with the plasmid in it,
Yeah.
and one had just E. coli.
Yeah. Yes. Yeah. And those were our testing
Okay,
trials. Yeah.
yeah, so then here, when we had 4 plates. And we put this one on 2 plates. And we put this one on 2 plates. So here was E. coli in this little Eppendorf tube. Everybody stay with me. We had E. coli in this tube, and we put our plasmid in this tube. We put just E. coli in this tube, plus broth, plus CaCl2, whatever else we added to this one. But we did not put the plasmid in here. So if we didn't put the plasmid in here, it's just E. coli. So if it's just E. coli, it's going to grow, would it grow on with no antibiotics?
Yes.
Yes. If it's just E. coli, will it grow with antibiotics?
No.
No, nothing. Okay, so here was our E. coli with our plasmid. So our E. coli with our plasmid, would it grow without ampicillin? Yes, it'll grow everywhere. If it's E. coli with our plasmid, will it grow on ampicillin positive?
Yes.
Only our transformants, right? Only the ones that have the plasmid, because with ampicillin, if it took this, if this ended up inside, then now it's antibiotic resistant, because that's the antibiotic resistant gene. So if ampicillin, what class of antibiotics is that in? The cillins, penicillin. So it's a cillin. So what kind of antibiotic is a cillin? Cell wall. That's what are we used it as an example. What was that? Beta lactam, yeah. So this is a beta lactam. So it's making beta lactamase. So what these transformants are doing is spitting out beta lactamase. So now these little tiny E. coli cultures can grow a little bit because there's a little bit of beta lactamase around there that blocked those beta lactam rings. Yeah, good review. Yeah.
The gene that we're inserting into the plasmid is what's included on that? I thought the glow in the dark is the gene that we're inserting into the plasmid, or is it already on the plasmid?
Great question. Yeah, it's already on the plasmid. So this is our plasmid, but this is the only funky part of the plasmid. So just ignore this just for 1 second. We need an origin of replication. Oh, there's one more thing I forgot, is you have to have a promoter. This one actually has to have a promoter and all restriction sites. This one has to have a promoter and restriction sites. So let's look at this, for example. So if we just took this piece, we have a promoter. What binds to the promoter?
The RNA.
RNA polymerase will bind to the promoter and read the ampicillin resistant gene and make beta lactamase. Are you guys with me? I know 1/3 of you guys are totally confused. Here's a piece, here's a gene, here's a chromosome. It's a tiny chromosome, it's all DNA. Part of the DNA, you have to have a section that can be replicated so that the plasmid will make more copies so that there's more and more. You have to have an origin, a replication. You also have to have an area called a promoter so that the RNA polymerase will bind, make RNA, and then once the RNA is made, there's ribosomes that will make the protein called beta lactamase. Now that beta lactamase will block the beta lactam. Yeah.
What's restriction sites again?
Yeah, great question. So restriction sites are how we got the gene in there in the first place. So they're those little areas that can be cut and inserted. Yeah, so that's how we're cutting and pasting. That's how we put stuff in there. So there's these restriction enzymes that will cut at the restriction sites. Now we can glue something inside of our plasmid. So you have to have the same here. In a wild type, in a normal bacteria, what would be here? What would be here? And what would be here? In this operon. It's there, I can feel it.
There's a cheerleader.
It's coming, somebody's got it.
There's a gatekeeper.
In the operon.
I can't remember what they're called.
There's an operon. Yes. Regulator that codes for the repressor. RPO. This is an operator. This is where the repressor will come and bind and block it so that the RNA polymerase can't transcribe. RPO, Rochester Philharmonic Orchestra. I couldn't think of another one. If you got another one, let me know. RPO. The promoter is where our RNA polymerase will bind. The repressor will bind to the operator so that the RNA polymerase can't keep going. This is a wild type. Why would we put a regulator in our plasmid? We wouldn't unless we wanted to stop transcription and translation. We don't want to stop transcription and translation, so we don't need those in our plasmid. We don't want to put in a regulator. We don't want to put in an operator. We don't need any of that. All we need is the place for RNA polymerase to bind and transcribe and translate our gene, which is ampicillin resistant. Yeah?
This might be kind of off topic, but if you were to do this in real life with what's it called, an insulin producing E. coli,
Yep.
then wouldn't that also kind of have do the E. coli have kind of like a lifespan of, they have like an expiration date, like you switch out your E. coli because they're going to start not producing it as high quality insulin?
Yes. Yeah, so you have to replate them probably every day, every other day.
Oh, wow.
Yeah. Yep, so people go in and replate them and keep the back stock fresh all the time so that they're producing insulin. They probably have some sort of chart about the temperature and the nutrients that they put in the agar and getting the right balance to where they're producing the most insulin in the least amount of time for the cheapest amount of money.
Is that to protect against mutation? And because obviously they're not going to be perfect every time.
Yeah.
Getting the right thing.
Yeah, I don't know how they they protect against that. I'm sure there are safeguards to make sure, and then also to make sure that the protein is high quality and that the protein is formed correctly and all that kind of stuff. I'm not sure how they would do that, but that's a good question.
So I'm just confused on, so in this lab, we were looking at whether or not they glow in the dark, right?
Yep.
So, but I thought whether or not they glow in the dark is how we assess whether or not the foreign gene was uptook by the plasmid. But-
How we assessed that it was in there, right? Uh-huh.
But if it's always in there, as well as the ampicillin gene's always in there, then what does that have to do with whether or not our gene was uptook by the plasmid?
So what we did, so on this plate.
It's not whether or not the plasmid takes it in, it's whether or not the bacteria takes in the plasmid.
I think, is that what you're asking?
Bacteria takes the plasmid.
Yeah, so some bacteria, let me draw it better. Some bacteria we put a bunch of plasmids in the Eppendorf tube with these bacteria. And we put all of these plasmids in there that we bought from the store into this Eppendorf tube. We put calcium chloride so it'll get closer, then we heat shocked them. So some will take up the plasmid, and some will not. So let's look at these two and compare the ones that took up the plasmid, that we induced competency, and some that we did not induce competency, that these ones died or they didn't take up the plasmid. So let's look at that really quickly. So here, we have no plasmid. They're gonna do what they would normally do. E. coli would normally die on a plate full of antibiotics, and they would normally live. They would die on a plate full of antibiotics, and they would normally live if there's no antibiotics. So we know that. Those are our controls. So let's look at our transformants.
That the plasmids have both genes. That's not the question.
What are those genes?
Well, the gene for the glowing and the gene for making insulin or whatever. Or not
For-
making
for
insulin,
green
beta,
fluorescent protein.
the gene for making beta lactamase.
Yeah, so we have two genes we're looking at. So here, if there's no antibiotic, this one is gonna live and this one's gonna live because there's no antibiotic. So that our transformants lived and our wild type lived, both lived. So here we have a plate full of bacteria, and we have some that glow in the dark, only a couple that glow in the dark, because they're all competing for resources. So these ones are gonna glow because of our GFP gene, and these ones don't glow, but they're both going to grow because there's no antibiotics. But let's look at this plate, we'll come back to that for a second. Let's look at this plate. So this plate, there is antibiotics, so if there's antibiotics, this one's gonna die. This is the only one that's gonna live. So we killed off the ones that did not take up our plasmid. The ones that took up our plasmid are the ones that are living. So these ones are the transformants that took up the plasmid. So these are all the glowing ones that took up our plasmid.
That makes sense. It's just more so that the gene we're inserting into the plasmid. Like, where does that come into play?
How did the gene get in there?
Well, why are we putting it in there?
So we're putting it in there just to see if we can, just to see if how to get a plasmid into a bacteria. We got it in there. Sorry, go ahead. Do you have a better way of explaining it?
Dr. Duffy, are you asking?
Like the specific one you're inserting between the restriction sites, not the plasmid itself.
The one that makes it glow.
No, the one.
How-
He's
Okay,
talking
the
about the
genes
one
in here?
labeled gene.
No,
Like,
not
how did we
that.
get these genes in here?
No, why are we putting that gene in there?
Why are we putting ampicillin?
No, the gene we're
GFP?
inserting, the other one.
Oh, this one.
Yeah.
Oh,
I guess
very
just a
good
diagram,
question.
And
Oh, no, this
how
is
do
a
we
very
know
good
we
question.
put that in there?
No, this is a very good question. So just ignore this for a second. Let's just ignore this. Ignore that. Let's just talk about these two for a second. Very good question, super good question. So here, we looked at ampicillin resistance because the ones that were not ampicillin resistance died for the most part. The ones with GFP, green fluorescent protein, survived because of the ampicillin resistance. So these are the only ones that are living on that plate. Hypothetically. Let's keep going with that for a second and I'll come back to what you're talking about. So what this bacterium is gonna do is they're making this beta-lactamase enzyme and they're spitting it out all over the place. This enzyme is everywhere because they're trying to protect themselves. So they spit out this enzyme all over the place. So because they glow in the dark and they're spitting out beta-lactamase because they took up our plasmid and we told it to do those two things. They glow in the dark and they make a glow in the dark protein and they make beta-lactamase. So these ones do. These little ones around here were just able to survive. There's E. coli around here that are not very healthy, that are trying to survive because there's beta-lactamase around here. So those are called satellite cells or satellite colonies. Does that make sense? Does the satellite part of it make sense? Yes? No? Thumbs up, thumbs down, medium? Okay.
You said the satellite colonies don't necessarily have the uptake of
Yeah, they don't.
Okay.
Mm-hmm.
Okay.
These ones are just making, so these
They're just
ones don't
lucky
glow in the
because they
dark.
have a good neighbor.
Yeah, they have a good neighbor.
Okay.
So they're like hiding under the umbrella of the ones, they're being protected from these ones. Yeah. So, okay, so that's ampicillin resistance, that's GFP. So what this is this is a hypothetical gene. And I probably shouldn't have drawn it on there, but this is the way we would draw a plasmid. Every plasmid that we would make, if it's insulin or whatever it is, we want to put in a gene over here. All we're doing in our lab is we're trying to see if it'll grow and trying to see if it'll glow. Like, what would be the point of that? Well, you want to put in a gene over here to make it do something. What are we making this plasmid do? You know, here we're just testing it. Like, did it work? Did it not work? But here, we're gonna, if we put in the insulin gene, now we can pick this colony, this colony, this colony, this colony, and they will start making insulin times a million. So so this is your gene that you're looking at, that you're experimenting on. This is just to be able to isolate, and this is just to be able to see if it worked. Over here is some functionality of it. We want it to do something now.
And if they took, so like every single transformant on the ampicillin positive plate, those all would have our gene of interest.
Yes.
So this is insulin.
Yeah. Yeah. Great questions. Yeah.
So just as a little reminder, when we did this lab, when we had the plasmids, were they already engineered to
Yes.
include both of those genes?
These two.
Yeah,
Yes.
so we didn't do anything, they
Nope.
just were already made
Yep,
that way.
we just bought it that way.
Okay.
That's all it had.
Okay,
on
so
it
all
like
we
that.
were doing is checking to see if we could get them into the bacteria.
Yep.
Okay.
Yeah.
See,
Yeah.
I thought the gene we're inserting made it glow.
Oh yeah. No, that's the GFP
So
gene.
those were already there.
The GFP was already there. The ampicillin resistance was already there. It already comes with the ORI. It already comes with the promoters. It comes with the restriction sites. That's how they built it.
So we
Yeah.
were just checking to see if we could get them into the bacteria.
Yeah. It was just a trial and error to see can we
How-
do it. And
How would you know if the plasmid uptook the gene you were trying to put in there?
because
Because couldn't it not up to the gene still glow and still be ampicillin resistant?
no, your plasmid, you would know your plasmid worked. Let me think about that.
See, I thought we knew it up to the gene because it was glowing, but the
Mhm.
glowing factor was already separate from that. It's already on the plasmid.
Yeah, so there's another way you can look to make sure your plasmids are whole and that they're correct. And the way you do that is something called electrophoresis, which I'm bummed we didn't do this section, but you would put your plasmid into this little electrical thing, and your electrical thing would say, this insulin gene is like 500 base pairs long,
Okay. So you're gonna measure the weight of it.
so you measure it. Yeah.
Okay, thank you.
Uh-huh, yes, yeah, thanks for clarifying, that's super helpful. Okay, whew, how do we feel about transformation?
What would be the hypothetical question that could be?
So definitely know what a plasmid is, what a plasmid looks like, the pieces of the plasmid, know, what the point is of putting it on the ampicillin plate. What the point is of putting the GFP in so that we could look at it under the UV. What's going on with those satellite colonies? So those satellite colonies aren't the ones that you would pick. You'd pick the transformants. Those ones are just surviving. Let me think of what else she's asked before. Competency, how to induce competency with calcium chloride and heat shock. Heat shock is the most important. I'm trying to think if there's anything else she's
What about
asked.
the number of transformants?
We heat shock, is that,
Yeah, I don't think she
What is that
asked.
like in the milk run?
that would.
What is the what, the milk one, yeah.
How would you answer? How would I?
Did we have transformation on the last one that we did? No.
Okay, so that's it. You guys can go through that again if you want to, but make sure you know what a plasmid is, know what the parts of that plasmid is, know how to induce competency, why we plate it on ampicillin, why we look at it under UV light, GFP, we're looking for GFP glowing transformants, also satellite cells are on there, that's transformation. Okay, let's do Kirby-Bauer. So Kirby-Bauer, one thing that's important that you guys need to know from Kirby-Bauer are your antibiotics. But that's funky is because those antibiotics from Kirby-Bauer are different. They're not different. They are, they're the same, but they are, let me see, Kirby-Bauer. I'll just leave them up there and you guys can go through them again. So, recognizing those antibiotics and what they do. So I'm gonna leave that up there for after I leave. Okay, so Kirby-Bauer, what is the whole purpose? What is the purpose of Kirby-Bauer?
To check for antibiotic resistance.
Yes, antibiotics, sensitivity versus resistance. So, 2, you can look at any bacteria. 3, we did procedure, we did the Mueller-Hinton. I have no idea if that's how you spell it. Mueller-Hinton. That's all. Oh, and then the discs, the antibiotic discs. That we flamed our tweezers. I don't know if that's important. I've seen something about flaming something at one question at one point, but methods. Anything else for the methods for that? No. Our reagents, we had no reagents. 5, we had results. So being able to measure and look at the chart. So you're gonna measure the diameter of the plaque of the clearing around the disc.
What is that clearing called? It had a name, didn't it?
A plaque, isn't it called a plaque?
What was the circle?
The whole thing, don't forget you have to do a lawn. I thought it was called a plaque, am I wrong? Yeah.
Just to make sure, since we also use a nutrient controller, would you have to let them know, like this?
No, Mueller-Hinton is what you use, yeah. Okay, that was easy, but that's not easy, so go through those.
Oh, for this one,
Oh,
should we know the different classifications of the antibiotics specifically?
So know this. This.
Okay, cool.
And what this was, if you guys remember what the clavulanic acid was, that was a beta-lactamase inhibitor. So we have a beta-lactam. Stay with me for a second, this is important. Beta-lactam is from the fungus. beta-lactamase is from the bacteria that cuts it. A beta-lactamase inhibitor is the chemical that we add to block the beta-lactamase so that it will still kill the bacteria. To block that little Pac-Man so that the Pac-Man can't cut the beta-lactam. Okay, let's move on.
I just found it. It's the zone of inhibition.
Zone of inhibition, awesome. Cool. Okay, ELISA. So we have a direct ELISA that we're looking for. Oh, let's do it the way we were supposed to do it. Let's do it 1 through 5. The purpose. What's the purpose of an ELISA? That's a good question. Yes, we're looking for,
Antibodies and antigens.
yes, antigen or antibodies. And a lot of times we will use this for viruses. We can use it for other things too. But we can use it, it's a common thing to use for viruses. Because viruses are so hard to find. Bacteria, we can plate it on an agar plate. Viruses, we can't find it. It's inside the cell, they're tiny, tiny, tiny. We have to look for our body's reaction to the virus. So here we're looking for antigen or antibodies. Our organism, we didn't do an organism, but we looked at HIV, we looked at Lyme, we looked at influenza, avian influenza, we looked at
West Nile.
West Nile. This is our only bacteria. This was a virus. What else? Methods. So direct, you look for the antigen. It's gonna stick to the bottom of the plate. Indirect, you look for the antibody. Let's draw those out really quick. So direct is going to be from the patient. Let me make the patient's a different color. So here's the patient's antibodies. I'm sorry, antigens. Here's our store-bought antibodies and our enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay. Here's our enzyme-linked antibody. And then we added a chromogen that will change color. I'll make these all, because we store-bought all of those. And then our indirect. These were our store-bought antigens. Then we have our patient's antibodies. Then we have an anti-antibody, or a secondary antibody with the enzyme and the chromogen. The green are patients, from the patient. The red is everything we bought. Yeah?
So it seems like direct is going to tell you if you're currently infected, and indirect is going to be more like if you've ever been infected.
That's a good point too, yes. Yeah. Okay, then our reagents, we can do antibodies, antigens, chromogens, things like that. And then 5 was results, was you can measure it in a titer form, so you can measure quantity, but also it'll be the color change will be positive. Okay, so fungi, protozoa, helminths, we're gonna go over today. Fungi, what are these? Yeast. What is this called? So this is a yeast that, like Candida albicans, yeast infection, thrush. What else? Yeast infection, thrush. Who else gets candidiasis? I can't remember. Candidiasis causes all kinds of things, but those are the most common. Then we have the sporangiospores. And then we had 2 types of conidiospores. They're both conidiospores, but how you can recognize the difference. This is penicillium. We'll look at downstairs. This is aspergillus. This is rhizopus. You don't have to know that. And that's candida. Or like, candida is our biggest yeast that we worry about, but there's other types of yeasts. We'll just say candida. These have this bigger like vesicle. You'll see it in the lab today.
Are the second 3 a type of yeast or are they molds?
These are all molds.
Okay.
And this one actually is a dimorphic, but just kind of forget that.
Okay.
This is mostly in yeast form. Okay, protozoa. We have these, let me not draw it that way. We have 4 types. We have this one that looks like a little banana slug. Then we have these ones. Then we have these ones. These ones. And these ones. Then we have these ones. And we have these ones. Okay, these are non-modal. Most likely Plasmodium. What does Plasmodium cause? Malaria. Awesome. This one with the little eyeballs is Giardia. Lamblia. That's the one with the little eyeballs, Giardia. That's the one, the 2 little eyeballs, they're suckers. And the reason I always remember that is this is what my mom had and it grosses me out a lot. Is that those 2 little suction cups suction to the walls, the lining of the intestines. So Giardia has those little suction cups. This is also called beaver fever. It's common among hikers, also in other countries. Giardia gastrointestinal. This one is also really disgusting. This is Trichomonas vaginalis. It kind of looks like a little fish. And that one is penile vaginal sexually transmitted. This long looking one is Leishmania. That causes a purulent pustular rash. It's aerobic, so it's on the surface. Sand fleas. Fecal oral. Insect, fecal oral, sexually transmitted, sand fleas. This one is trypanosoma. This one looks like, trypanosoma looks like more like a helmet, kind of. I'll show you in the microscope today. This one is from the sleeping sickness, from the tsetse fly and the kissing bugs. This one is B. coli that has the cilia. This one is an amoeba, like Entamoeba histolytica, that's amoebic dysentery. Fecal oral, bug, sexually transmitted fecal-oral bug. Okay, helminths are hard to look at in the microscope, but we had these ones. Then we had these ones. Then we had these ones. Then we had these ones. We had this one. And then we had whipworm. Work with me. These ones are which ones? With the proglottids. Anyone remember?
Intestinal.
Cestodes. These are both flat. And this is a trematode that has a snail vector. These are like flukes. Like leeches. This is a roundworm. These are all roundworms. This one's called Ascaris. These are pinworms. This is a whipworm. The other one is, you always see it inside of someone, but here it has little teeth. This is a hookworm. These are all round. We'll try and see what we can see today, but it's hard to see because they're, in the microscope, they're giant, so you wouldn't need a microscope. Okay, I'm gonna go over one more thing. We're gonna do it together. Is conversions. Let's do this together. 1.5 times 10 to the negative 2 milliliters. What pipette would you use? Micropipette. Well, micropipettes are microliters, so we have to change it from milliliters into microliters. How do we do that? We do the train. Here's our train. We have milliliters. We want to get rid of our milliliters, so we're going to cross those out. We want microliters. So the microliters are going to be on top. We have milliliters, milliliters. This is the way our train is set up. Here is what we have. 1.5 times 10 to the negative 2 milliliters. Here, this is what you have to memorize. This is it. So here, how many milliliters, how many microliters are in a milliliter? Well, we look here. Here's our milliliters and microliters. We already have it written down. This is the only thing we have to memorize. 10 to the third to 1. 1 milliliter, 10 to the third microliters. So now we just, dividing by 1 means nothing. We can get rid of that. All we're going to do is multiply these two. 1.5 times 10 to the negative 2 times 10 to the negative, or 10 to the third. Here, we added a 10 to the third here. So here now we just add these. 3 plus a negative 2 is 1. What this 1 tells us is how many times to move this decimal. 1 is saying move your decimal once. If you're moving it negative, you go that way. If you're moving it positive, you go that way. I'm right-handed, positivity means right. I'm left-handed, negativity means left. My left is useless. My right is strong. My right hand, you go this way, positive, negative, you go left. So here says 1, I'm moving it over 1. I moved it this way 1 time. I'm left with 15 microliters. So what am I going to look for on the top of my pipettes? If I have a P10, I have a P20, I have a P100, I have a P200, and I have a P1000. Which one for 15 microliters am I going to use? 20.
What does the 20 mean?
P20, that's 20 microliters maximum.
That's the max amount.
That's
okay.
max.
In microliters.
In microliters.
Okay.
All of them are max in microliters. Great question. Okay, so here it's gonna give me another option. Well, is it 1.5? This is option A. This is option B, for example. 1.5 times 10 to the 0. 0 tells me I moved the decimal 0 times. I can write it out here, whatever you want. 1.5 times 10 to the 0 milliliters. I want microliters. I have milliliters on the bottom. I'm trying to get rid of milliliters. Microliters left. 10 to the third. So 1.5 times 10 to the 0 times 10 to the third. 3 plus 0 is 3. 1.5 times 10 to the 3, it's positive. Positive is right for me. So if you have 1.5 and you move the decimal over 3 points, 1, 2, 3, you have 0, 0, it's 1,500 microliters. What one would I use? None. That's not right. No. There's not one.
You'd have to do it twice.
Yeah, you'd have to do it twice.
You'd have to use a 1,000 and then 500 of the 1,000.
Yeah, so if this was an option on the quiz, you'd know it's not that. So you have to do this for every answer to figure out which pipette it matches. So it seems like a lot of work, but what you can do is on your scratch paper, write out the possible solutions and then try and work it out on your scratch paper when you're done. You know, like, okay, now I'm gonna work through it one more time. I'm gonna go through and do my conversions for every answer. And then I can pick which one it is. So then I have more time, I can think about it later. You can also come back to it later. Yeah?
How much time do we have on the lab
I think we have unlimited time, really. Because last time we had 90 seconds per station. I gave you guys about 10, 15 minutes. If you guys need more than that, you can take more than that. Would you guys, this is optional too, would you guys rather have 2 minutes at each station? Or 90 seconds? 2 minutes? 2 minutes? Yeah, let's do 2 minutes each station then, and then if you still need more time at the end. If you feel like, and you know, I think it might be a good idea too, because I've seen her double up a lot of the questions this time, so I think there's more questions per station. So yeah, I'll give you guys 2 minutes, but then at the end, if you still need more time, if you're like, I still need 3 more stations I need to check out, just let me know, no problem, totally fine. Yeah? Oh yes, and I will leave all this up here, you guys can check it out. So it's 10 to the third milliliters equals 1 liter. 10 to the third microliters equals 1 milliliter. 10 to the sixth microliters equals 1 liter. So that's all you have to remember. But also if you just add these two together to make 1 liter, that would make sense too, but that's fine. Just do that way. So when I'm gone now, it might be a good idea if you guys feel confident, if you can MR instead of deciphering all the things with MR, put the reagents, put what you're looking for. Carbohydrate fermentation, I'm looking for an acid product. Carbohydrate fermentation, I'm looking for a neutral product. What were my reagents? Citrate, what was I looking for? Go through those quickly for those funky ones, the easier ones, and then spend a little bit more time on plate count. Spend a little bit more time, maybe like INVIC, all the pieces of the INVIC. Yeah? Okay. So I will be down there. You guys can just come when you're ready. Our goal is to be leaving by 4:30. So I'll just start pulling down the microscopes at 4:30. But hopefully you guys can just kind of run through, have Dr. Roseberry check your book, you know, make it kind of really mellow for the microscopy downstairs. Spend some time on this together. Okay, so I'm going to leave all of this here, and then are you guys gonna stay here? Would somebody mind erasing the board for me? And then, do you mind if I show you just how to shut this off? Do you mind shutting this off for me? And just, if you wanna leave before everybody else, just tell them how to do it.
Okay.
Just push that button, that's it. That's all you have to do. Oh, you know what, actually, you have to push this button and this button.
Okay.
And that's it. Awesome, thank you so much. Okay, and then if you do wanna leave, just tell somebody else. Okay, cool. So somebody be brave and come on up and take turns. Don't leave all the burden to one person.